Today we're going to be talking about viruses. Now, I wanted to emphasize a couple of things that are related to topics I think that we've already talked about in class. And this is having to do with how viruses actually get into a cell. So this blue thing down here is going to be our cell. And just for the purpose of relating this to real life, make it more applicable, let's say that this is a human cell. And the way that a virus gets into a human cell is by binding to proteins on the human cell surface. So we have lots of proteins on our cell surface, as we have talked about in the past. They have functions such as identifying the cell to other cells in the body, as well as some of them are channel proteins, so they let things into the cell or they let things get out of the cell that need to leave the cell. So we have lots of different proteins in the cell plasma, in the cell's plasma membrane. Again, we'll say this is a human cell. Now, viruses, as we saw in the reading, are going to take advantage of these proteins that are on the surface of the cell. And viruses can bind to these proteins, but it's very specific as to which protein a virus is going to bind to. And this is why we talk about the different strains of the flu, and we've talked about the different strains of coronavirus. So let's say this is our virus. It is an enveloped virus, so it has a plasma membrane. And then these are the spike proteins, little things sticking out that I just drew. And the spike proteins must be able to fit with the shape of the protein on some protein on the cell's surface. So here I've drawn it so that these spike proteins can match up to this protein on the human cell surface, and it should be able to bind. And then once the virus has bound to the cell's surface, it will be able to subsequently get into the cell. So there are other mechanisms used to get the virus into the cell. Oftentimes it's like a little type of kind of a machine or almost a cascading effect. Once it's bound to this protein, then it's going to get inside the human cell. Now, let's say that instead of a human virus, this virus is a virus that mainly infects chimpanzees. And if this virus mainly infects chimpanzees, then maybe it will have a similar spike protein to the one in humans, but it's not exactly the same. So instead of having that V shape, maybe it has more of like a curved shape. And because that shape doesn't really match up to our human cell proteins, we will not be infected by this virus. So this virus is not going to be able to bind to this human cell. It's not going to be able to infect it because binding needs to occur before infection. But the danger is, so normally we assume that a virus for some species cannot infect another species, but the danger is that we can have a mutation in that spike protein. Keep on changing color for the eraser, which is not helpful. So we could have a mutation. So maybe this becomes a little bit less curved. Maybe it's still not exactly the same as the protein that we saw previously, but it's a little bit more similar to being able to match up to the human spike protein or the human surface receptor. And again, students tend to ask, wait a minute, why do humans have these receptors on their surface that let viruses in? They have some other job. The receptors on the human cell surface are there to identify the cell to other human cells or to do some other job. They're not there to let viruses in, of course. So if the spike proteins on the virus are somewhat similar to matching up to human cell surface proteins, then a mutation might make them similar enough that they're able to bind to the human cell, and then another mutation in the future might make them even better, etc. And so then you have suddenly jumped species. Okay, so how do these species jumps actually occur? And how do we have some viruses that are able to infect multiple types of organisms at the same time? So for example, we are all familiar perhaps with the West Nile virus. And West Nile is a virus that infects mosquitoes, birds, as well as humans. Please excuse the messy writing. And the reason why some viruses are able to infect different types of organisms at the same time is because of the similarity of the proteins on the surface of the host cells. So mosquitoes and humans share some similar surface proteins. On some of their cells, not all of them. But if a virus is well adapted to bind to one of these, then it may be able to bind to the other as well. Now, the reason why surface proteins can be very similar between different species is because they're just very good at their job. The proteins work very well, and so it would be disadvantageous to change them. Another possibility is with the H1N1. So think about H1N1, and if you recall, our H1N1 virus has more than one type of spike protein. So these are obviously not very accurate, but I've drawn two different types of spike. One is blue and squiggly, the other one is green and straight. And so these two different types of proteins on the surface of the virus might be able to bind to one cell surface protein in one species, and then the other one might be able to bind to the other species. So maybe the blue one is able to bind to cell surface protein in mosquitoes, and maybe the green one is able to bind to a cell surface protein in humans. Again, I'm not saying that's true for this specifically, but these are the possibilities. Also interesting in terms of species or host species and viruses is that we know that many diseases, maybe not many, but at least some significant diseases, have come from other species. So for example, the now more or less eradicated smallpox virus actually came, we believe, from camels. Camels. Oh boy. Okay. So how do we figure this out? Well, smallpox and camelpox, well, you guys know what I mean. Smallpox and camelpox, when you look at their genomic sequence, it is very similar, and that could mean a couple of different things. It could be coincidence, because they both infect similar types of organisms and they happen to use the same type of cell receptor, so that means that their genetic code to make that receptor would be similar to one another. But it could also mean that they are actually related, and that we either got smallpox from camels or vice versa, but the history shows that camelpox was around before smallpox, most likely, and so it seems more likely that we contracted camelpox after some type of mutation in the virus and it became smallpox. And so, although these species jumps are pretty rare, when they do occur, they could become very dangerous. And especially because there are certain hosts that are much better at transmitting disease than others. And so mosquitoes are a really big one, right? Mosquitoes transmit many different types of disease and many different types of virus. And one of the problems with mosquitoes is not only that they are carriers for many different types of disease, but they are also excellent hosts for disease. Now, what does it mean to be an excellent host? Well, it's not like hosting a dinner party, but actually in a way it kind of is, because the virus has an excellent time infecting mosquitoes, and viral load, which is the size of the population of virus in the host, is going to be very high. So there are lots and lots of viral particles that are produced in a mosquito, especially in terms of things like West Nile. On the other hand, the viral load of West Nile in humans is fairly low, especially compared to in the mosquito. So the population size, even though it can affect an individual a great deal, the population size is relatively small compared to the population size in a mosquito. So the human for West Nile is actually a dead end host because the viral load is low enough that it's very unlikely that that virus is going to be passed on to either another mosquito or directly to another human. And so that's what we call person-to-person transmission, and we do not have that for West Nile, at least not right now. Again, that can change in the future because these viruses can evolve fairly quickly.